Very good. I'll do a casual introduction of you, Dr. Kushwah.
Okay.
You are, as our website states, an accomplished scientist, which we know, and co-founder and chief scientific officer of Anam Floyd. Did I pronounce that correctly?
Yeah, got it right.
A leader in clinical genetic testing. Very true. Vast experience in different areas of human diseases, particularly stem cells, genetics, immunology, and regenerative personalized medicine. Dr. Kushwah held medical faculty and scientific appointments at U of Ottawa, the National Research Council of Canada's Human Health Therapeutics Branch, and a hospital for sick children in Toronto. He received numerous and prestigious awards by the Canadian government and several foundations. He's a regular speaker in international circuits, a review editor for several medical journals, and author of numerous peer-reviewed articles. Rahul currently teaches Understanding Genetics for Improving Health Outcomes at the Institute of Holistic Nutrition in the Continuing Education Department, and he is here this evening to give us an excerpt from the course. Thank you so much. We know you could be doing many things this evening, but you are with us, so thank you.
Thank you for the wonderful introduction, Julia, and thank you everyone who's joining in. So let me just share my screen first for the presentation. So today our focus is going to be on the role of genetics in nutrition and detoxification. So perhaps the biggest discovery in the field of medicine over the last few decades has been the mapping of human genome. And what that has led to is identification of how genes control our diet, our nutrition, our ability to detoxify. And what I'll be talking about is how you can take genetic information of an individual and how you can utilize it to offer what we call personalized healthcare. Because each and every one of us has some unique biological phenotypes because of the underlying changes in DNA. And it's only when you target the underlying DNA-based information that you can have personalized healthcare, which can maximize efficacy. So when it comes to the Human Genome Project, it was initiated in 1990 and the first draft was completed in 2001. So definitely it was a very long and cumbersome process that involved a lot of scientific teams. But at the end of the day, what we got was a complete sequence of the entire genome. And that really laid the foundation to what we refer to as personalized healthcare. For instance, we have genetic differences when it comes to metabolism of nutrients or absorption of nutrients. And as such, the nutritional needs of one individual is going to be different from the other because, again, they have underlying differences in their DNA. And that is the basis for personalized nutrition based on DNA. Similarly, when we talk about fitness, we have underlying differences when it comes to our muscle genotype and muscle physiology. And as such, you cannot really have the same exercise regimen work on every single individual. And that's why the mapping of human genome laid the foundation for what we refer to as personalized fitness. Along with that, a very important field is that of pharmacogenetics. So when you talk about any medication, the efficacy hovers around 40%. So what that means is that only 40% of the population is going to respond to a medication. And at the same time, you'll see a big chunk which will have no response. And of course, a significant portion of people out there that will show an adverse reaction. But again, all that is coded in your DNA. If you understand the genetic information of an individual, you can pretty much identify what medication is going to work for the individual and what medication is going to give them a serious adverse reaction. Similarly, when we talk about disease risk, perhaps the most common example is when we talk about breast cancer and BRCA1 gene or BRCA2. So individuals that are carrying some variants of BRCA1 or BRCA2 could have an up to 90% chance of developing breast and ovarian cancer. And again, that is something which can be identified by looking at genetic information or DNA of an individual. So when it comes to genetic testing, I mean, it's all based on reading the DNA, which stands for deoxyribonucleic acid. And we can simplify it as calling it the life molecule or the molecule of life. So when it comes to eukaryotic cells, which are cells that are found in almost every multicellular organism, including humans, there is a structure in the middle, which we refer to as a nucleus. So think of it as a small cell being in a bigger cellular structure. And inside the nucleus is where DNA is present. And DNA by itself is wrapped around a bunch of proteins, which are called histones, to generate condensed structures, which are referred to as chromosomes. So when it comes to humans, we have 46 chromosomes. We get 23 chromosomes coming from the maternal side and the other 23 from the paternal side. And when it comes to the chemical composition of DNA, DNA is really a sugar molecule. So what you have is a sugar backbone, which is joined by phosphodiester bonds, and at the same time, you have specific molecules which are referred to as nucleotides. And you only have 4 versions of these molecules, which is A, T, G, and C. And when you look at 3 of them together, such as if you look at the picture here, and if you look top down, for instance, if you were to read A, G, and T together, that is referred to as one codon, because that combination by itself codes for one amino acid. Now, when it comes to the hierarchy of DNA, of course, first you have your chromosome, then you have something which we refer to as a gene. So a gene refers to a section of DNA that codes for a protein. And again, the key is to understand that not every single DNA molecule codes for a protein. So it's only a subset of the genetic information or the DNA that we have which codes for proteins. And it's only that segment of DNA which is referred to as genes. In terms of human genome, almost 30,000 different genes have been identified so far. So how does it all work when it comes to going to protein from DNA? So DNA is found in the nucleus. From DNA, you get a messenger molecule, which is referred to as the RNA. And then there are certain proteins that bind onto the RNA that read the codons, and based on the codons, they assemble amino acids together, which gives generation to the protein. And how do mutations impact all that? So on the right here, we have a very simplistic diagram. And what it's really showing is, so let's say you have a normal strand of DNA. And that's the strand which is shown right here in the middle. So normally this DNA strand codes for a protein, which can be an enzyme. And the function of this enzyme is to take this circular substrate and let's say it turns it into a square. But for it to do that, the circular substrate has to bind to a pocket within the enzyme. And that's really a chemical binding and a chemical reaction that happens at the end of the day. But again, from a 3-dimensional standpoint, what you really need to understand is that for this round molecule to bind into the enzyme, there has to be the right pocket. So let's say now if there's a mutation in this gene and it impacts the structure of the pocket, what could potentially happen is that now the circular substrate or the molecule is not going to bind to the enzyme. And the next thing you know, you don't have any more of these square product of the enzymatic reaction which is produced anymore. So effectively, by introducing a single mutation in your DNA, what you have resulted in is a mutated enzyme which has completely lost its functionality in terms of whatever the enzymatic reaction there is. And this is really the basis for what we refer to as nutritional genetics. Now, when it comes to any nutrient, it can be simplified as a 2-step process. First and foremost, the nutrients have to enter into a cell, and they do so through nutrient transporters, which are, of course, proteins that are coded by DNA. And with a lot of these nutrients, once they go into the cell, they have to bind some very specific enzymes which convert them into the bioactive form. And it's the bioactive form which is the only form that can be used by the cell. So again, the key to remembering here is that when it comes to the nutrient transporters or the enzymes, both are encoded by DNA. So let's say if somebody has a mutation here in nutrient transporter, what's going to happen is that you can give them as much as you want of this specific nutrient, still they will be developing deficiency because this nutrient will not be able to enter the cell. On the flip side, let's say there is another individual where they have a mutation in this enzyme. Even though the nutrient is going to be entering the cell, it will still not be converted into the bioactive form, and still they will have deficiency. As such, for those individuals, you'll need to provide them with the bioactive form of the nutrient. But again, to have answers to these questions, you have to look at the genetic information of the individual. Now, when it comes to the nutrients for which a clear genetic link has been identified, they include vitamin A, B6, B12, choline, vitamin C, D, E, folic acid, calcium, iron, magnesium, selenium, and zinc. Now, I'm going to go over two very simple examples that, again, illustrates the importance of why you need to understand someone's DNA before you prescribe them with a nutritional plan. And one of those examples is that of vitamin A. So when it comes to vitamin A, there are really two main sources of the vitamin. One is beta carotene, which comes from plants, and the other one is retinol, which comes from animals. But the key is for beta carotene to be used by our body, it has to be converted to retinoic acid. And this depends on an activity of an enzyme, which is referred to as BCO1. But again, you got to remember, any enzyme, any protein in the body, it's encoded by your DNA. So if someone is carrying a certain mutation, it impacts the underlying physiology. Now, when it comes to BCO1, it turns out that almost 45% of the population is carrying a mutation which is impacting the enzymatic activity. What that means is that 45% of these people, you can be giving them all the beta carotene that you want, but their body will not have the ability to convert it to retinoic acid. So again, it's illustrating the same point, which I said earlier, that there are enzymes which play a role in turning a nutrient into the bioactive form. And if there is a mutation, you can provide them with the specific form as much as you want, but still the individual is going to be developing deficiency. So what does this all mean? So what this means is if someone is carrying the mutated form of BCO1, which is the T allele variant, you can give them all the beta carotene that you want, but still they'll go on to develop vitamin A deficiency. But again, if you know beforehand they have the mutation, what you have to give them is a diet which is rich in the animal form of vitamin A or which is rich in retinol. But to address this, you have to understand the genetic makeup of the individual. The other example I'll be talking about is that of folate or folic acid. So of course, as you all know, it's an essential nutrient. Deficiency can lead to anemia. And in pregnant females, it can also lead to neural brain defect in babies. But when it comes to folic acid, for it to be used in our body, it has to be converted into a molecule which is 5-MTHF or 5-methylfolate. And the conversion of folic acid or dietary folate to 5-MTHF depends on an enzyme called MTHFR. And there is, in fact, a very high frequency of MTHFR mutations in the population level. So rather than going into the specifics of how the genotype is distributed at a population level, the point that I want to make is that almost 30% of the individuals out there are carrying a mutation in MTHFR enzyme, which is reducing the activity by 75%. So in other words, to these individuals, you can give all the folic acid or dietary folate that you want, but still they'll go on to develop folic acid deficiency because their biology or their body lacks an ability to convert it into the bioactive form. As such, for these individuals, you have to give them methylfolate. But again, to have this conclusion, you have to look at the genetic information of the individual. So overall, when it comes to nutritional genetics, it can help identify that for which nutrients someone has a very high chances of developing deficiency for. It can also clearly assist in developing a nutritional plan to prevent such deficiencies based on the genotype or the DNA makeup of the individual. At the same time, genetics can be used to identify which form of nutrients are going to be absorbed by the individual and what will not work for them. But the key point is that because we have these underlying differences in our DNA, you just cannot have one plan that's going to work on all. Because we have underlying genetic differences, and as such, when it comes to nutritional planning, it has to be based on the genetic information of the individual. Now, again, changing gears a bit, the next segment I'll be going over briefly is that of weight loss and genetics. So usually when a generic weight loss plan is given out, it involves two different components to it. One is, of course, diet, and the other one is fitness. But there usually tend to be three different outcomes that for some individuals, you see no weight loss. For some, you see slight weight loss, and for some individuals, you see dramatic weight loss. Why is this? Why isn't it that you give this generic weight loss plan and everyone just loses weight? Well, the answer lies in genetics, because people have underlying differences in their genetic makeup. And that's why they don't respond in a similar fashion to a generic weight loss plan. And from a dietary standpoint, perhaps one of the most famous aspects I can talk about is that of monounsaturated fats. So when you look at popular media or, again, a lot of fitness shows, what they always tell you is that if you eat a lot of monounsaturated fats, that will expedite weight loss. Yes, it does happen, but again, it only happens in a certain percentage of population. But again, it does not help a lot of people. With a lot of people, you can increase the consumption of monounsaturated fats and still they do not lose weight. Why is that the case? It boils down to a specific gene or enzyme, which is referred to as PPAR-gamma2, which plays in metabolism of fat and weight loss. Now, this is taken from a study published a few years ago, and here what the authors did was they looked at the genetic makeup of the individual in terms of PPAR-gamma2 status, and they could stratify individuals as having either the C allele, which is shown here in the dark graph, or in the black graph here, or they could have the G allele, which is shown here in the gray graph. And they gave them two different sets of diets. One set had a diet which had less than 56% of the fat as being monounsaturated fats, and the other segment had over 56% of their dietary fat as being monounsaturated fats. And what the study found was that it's only the individuals that were carrying the G allele that actually had a significant weight loss when they went on a diet rich in monounsaturated fats. Whereas individuals that carried the C allele, even if they were given a lot of monounsaturated fats, they did not lose weight. So again, what the point that this really illustrates is that for any weight loss planning, you have to understand the genetic information of the individual, because there is going to be a segment of the population that will respond to the plan that you're coming up with and a segment which won't. But if you understand the DNA makeup of the individual, you can have a plan which really addresses their biology and thereby maximizes efficacy of the weight loss plan. From the fitness standpoint, of course, the consensus is if you want to lose weight, you do a lot of strength training because that'll promote muscle gain, you'll burn a lot more calories, and you will have weight loss. But it turns out this does not work for everyone. A lot of people actually go on to gain fat instead of muscle if they do a lot of strength training. And the answer to this lies because we have underlying differences when it comes to our DNA for a specific gene, which we call INSEC2. So the graph here is taken from another study where they stratified individuals based on their genotype of INSEC2. So again, there are individuals who had the G allele and the individuals who had the C allele. And in the study, what they found was that individuals who had the C allele of INSEC2, when they did a lot of strength training, they had an increase in their subcutaneous fat on their biceps. In other words, they were actually gaining fat and not muscle. So what it means is that if you look at the genotype of the individual, and let's say you identify they're carrying the C allele, then in those individuals, you do not want to have a fitness plan which has a big focus on strength training, because these individuals are very likely to gain fat instead of muscle with strength training. This little snippet here was taken from Wall Street Journal, again, 10 years ago. And the summary of that article in Wall Street Journal was that if you give a generic diet to people to help them lose weight, on average, they lose around 4.6 pounds. But if you give them a diet which is based on their DNA, they lose over 13 pounds, so which is 3 times more. But again, the key message here is, if you prescribe somebody a generic weight loss plan, you'll usually see 3 different outcomes, which is no weight loss, slight weight loss, or dramatic weight loss. But if you can prescribe a DNA-based weight loss plan, the only outcome that you're very likely to see is that of dramatic weight loss. So when it comes to weight loss genetics, by understanding or looking at someone's DNA, we can identify what kind of dietary interventions will expedite weight loss. We can identify what dietary interventions will not work for the individuals in terms of promoting weight loss. We can again identify what kind of exercises will help the individual lose weight and what will not work on the individual. But at the end of the day, you have to look at the underlying genetic makeup of the individual to have a weight loss plan which maximizes efficacy when it comes to losing weight. Now, the next part, I'll be focusing briefly on caffeine. So of course, when I do this in class, I always ask the question that how many people think that coffee is good for them or coffee is bad for them. And a big chunk of people actually go out to say that they think coffee is completely bad for them. And the real answer is that there is no yes or no when it comes to coffee or caffeine. Caffeine can be a medicine to a certain set of individuals, and it can be poison to others. And the missing link there is, of course, the DNA or the genetic makeup of the individual. And the specific gene which plays a role in terms of identifying if caffeine is good for you or bad for you is a gene which is CYP1A2, and it's part of the cytochrome P450 complex. So when it comes to caffeine, caffeine by itself leads to vasoconstriction or constriction of your blood vessels, and it does so by suppressing the adenosine pathway. However, the moment caffeine is broken down by CYP1A2, you actually get constituents such as pyraxanthine, theobromine, and theophylline, and these individual constituents are potent vasodilators. In other words, they relax your muscle cells, they relax your blood vessels, and your blood pressure drops. So with caffeine, it's going to shoot your blood pressure up, but if caffeine is broken down and you get these constituents, it's actually going to bring your blood pressure down. And the missing link between the two is CYP1A2. So if someone has very high activity of CYP1A2, then caffeine will be broken down in their blood rapidly. But if someone has low activity of CYP1A2, then caffeine is going to stay in their blood for a long time. And whether CYP1A2 is working at a fast or a slow pace depends on one single mutation in the DNA. So individuals that are carrying the 1F allele of CYP1A2, they have reduced activity of this enzyme. So in these individuals, caffeine is not broken down rapidly, so caffeine can stay in blood for a longer time period. And in these individuals, what caffeine does is, of course, it narrows blood vessels and their blood pressure goes up. So for these individuals, caffeine is a big no, because it can lead to hypertension. In contrast, individuals that are carrying the 1A allele, in these individuals, the enzymatic activity is very high. So the moment they consume caffeine, it's broken down very rapidly into the beneficial constituents. And in these individuals, caffeine can actually help lower their risk of hypertension. So the chart here on the left is, again, it's based on data from a few thousand individuals. And what it really is saying is, so let's say if someone is a slow metabolizer, or if someone is carrying the 1F allele, if they were to have over 4 cups of coffee a day, they're increasing their chances of hypertension by 3-folds. On the flip side, if someone is a fast metabolizer, that is, they're carrying the 1A allele, and if they were to have over 4 cups of coffee a day, they're reducing their risk of hypertension by 3-folds. So that's why there is no straightforward answer to whether a food constituent is good for you or bad for you. The answer lies in your genetics. What's poison for one can actually be medicine for the other person, but you can only identify that if you look at the DNA makeup of the individual. Similarly, when it comes to lactose intolerance or gluten intolerance, or predisposition to lactose or gluten intolerance, of course, it's all underlying genetics. So when it comes to lactose, it's broken down by the enzyme lactase, and individuals that go on to develop lactose intolerance, they actually have low activity of lactase. And the way how it's regulated is through a secondary gene, which is referred to MCM6. So MCM6 actually has a positive feedback mechanism on lactase enzyme. So individuals that have increased activity of MCM6, in those individuals, MCM6 promotes production of lactase, and those individuals do not go on to develop lactose intolerance because they have lactase persistence. On the flip side, individuals that are carrying the G allele of MCM6, in these individuals, MCM6 has low levels of activity, so they do not have a lot of lactase production, and these individuals are at a very high risk of developing lactose intolerance. So when it comes to dietary genetics can help us identify if someone is at risk of lactose intolerance or gluten intolerance. It can identify things such as, is coffee good for you? What are your chances of developing sodium-associated blood pressure? Because again, not everyone who has a lot of sodium in their diet goes on to develop hypertension. Similarly, genetics can help identify what is the risk of an individual to develop insulin resistance with starch intake, things like if someone has a sweet tooth, their tendency of snacking. And of course, when it comes to whole grain, we tend to think normally that whole grains reduce our risk of diabetes. They do so, but again, not in every individual. But by looking at the DNA, you can get some specific answers when it comes to your diet. And with that, you can do DNA-based diet planning for the individual to help them improve or perhaps sustain their health. For the last part, I'll be focusing a bit on detoxification. So when it comes to detoxification, all of the detoxification in our body takes place in the liver. So that's where all the chemicals, alcohol, drugs, pollutants, insecticides, hormones, medicines, they're all broken down. And it happens through the cytochrome P450 complex. So when it comes to detoxification, it's really a two-step process. In the first step, which is phase 1, this is where you have activity of the cytochrome P450 enzymes. There is chemical modification of whatever is the compound which is being eliminated from the body. And that could be things like pollutants, medicines, and hormones like estrogen. In phase 2, there is conjugation to water-soluble molecules such that this compound, such as estrogen, for instance, can be eliminated through the body by becoming water-soluble, because once it's water-soluble, it can be eliminated through urine, bile, or stool. But when it comes to the detoxification pathway, not only do they play a role in terms of our predisposition to a lot of chronic diseases, but they play the main or the essential role when it comes to pharmacogenetics, because every single medicine that goes in our body goes through the detoxification system. And perhaps the best example I can talk about is that of codeine. So when it comes to cough syrups, I mean, of course, you see a lot of off-the-counter cough syrups at Shoppers Drug Mart or any pharmacy, but the reality is none of them have shown any efficacy in any clinical trial whatsoever. The only thing that has shown efficacy when it comes to cough is codeine. But the biggest problem with codeine is that there is an enzyme in our body which can rapidly convert codeine into morphine. And that enzyme is CYP2D6. But of course, individuals have genetic variation in terms of their CYP2D6 activity, where some individuals can have low levels of CYP2D6 activity, and some can have high levels of CYP2D6 activity. Individuals that have increased CYP2D6 activity in these individuals, even if they have a little bit of codeine, it's rapidly converted to morphine. And right away, they start showing symptoms of morphine overdose. So of course, these individuals should avoid codeine altogether. And again, this holds true for every single medication that you can think of. And that's why when it comes to genetics, it plays the central role in terms of identifying what medication is going to work on you, what will not work on you, and what is likely to give you serious adverse effects. And when it comes to diseases, perhaps the most common one of all involves estrogen and breast and ovarian cancer. So estrogen, of course, is produced by ovaries. It promotes proliferation of breast tissues. So overall, it can help induce formation of cancer. Our detoxification system is designed to clear estrogen, but again, in the process of detoxification of estrogen, there are cancerous metabolites that can be produced. For instance, if you look at the schematic here, if someone has high levels of CYP3A4 activity, then estrogen preferentially will be converted to non-cancerous forms. But of course, if someone has high levels of CYP1B1 or CYP3A5 activity, then estrogen will be preferentially converted into cancerous forms. But again, even after you get these cancerous forms, we have these GST enzymes that play a role in inactivation of these cancerous forms. So the example we'll be talking about today is that of the underlying genetics of GST M1 enzyme. So GST M1 enzyme is involved in detoxification of electrophilic compounds, just carcinogens, of course, estrogen metabolites, and it does so by conjugating it with glutathione. So individuals that have impaired GST M1 activity, in these individuals, they are likely to be at a very high risk of developing cancer simply because their ability to remove a lot of these cancerous metabolites is actually impaired. But again, just because somebody has impaired activity of GST M1, it does not mean it's the end of the world, because all of these detoxification enzymes can actually be induced, but to induce them or to suppress them is something you can only do once you understand the complete genetic makeup of the individual. So let's say when it comes to GST M1, what studies have shown us that if there is a dietary intervention with resveratrol, which is the flavonoid found in red wine, that can actually boost GST M1 activity. And there is clinical evidence to support it. But again, you would only want to induce GST M1 activity in individuals who have low activity of GST M1, and there is an underlying health risk because of that. So what does it all mean? So we can look at the genetics of an individual when it comes to the estrogen metabolism. We can identify if genes that are involved in clearance of estrogen metabolites have reduced activity. And if so, then we would want to increase the activity of the enzyme to make sure estrogen can be rapidly cleared from the body. On the flip side, by using genetics, we can also identify if some of these enzymes have increased activity, such as CYP3A5, because if there is increased activity of CYP3A5, you can get more cancerous metabolites coming out of estrogen. So in that setting, you would want to have a dietary intervention to reduce the activity of the enzyme. But overall, by looking at the genetic makeup of the individual, by seeing how their detox pathways are functioning based on their genetics, you can have an overall plan which reduces the risk of the individual from developing things like breast and ovarian cancer. But again, the key is understanding the genetic makeup of the individual, because you cannot have a generic plan when it comes to estrogen metabolism or detoxification or pharmacogenetics, because different individuals are going to have varying levels of activity of each and every enzyme. So induction of one enzyme for individual A may be beneficial, but again, the same induction for individual B could have an adverse effect. And that's why you have to completely understand the detoxification genetics before you come up with a health plan to reduce risk of a specific disease for the individual. Now, this is taken from one of our sample reports. So again, if we were doing it in class, I would have asked you guys to give me an answer to this question. Overall, it's about seeing that, let's say if you were given a report whereby somebody had normal CYP1A1 activity, increased CYP1B1, decreased CYP3A4, normal COMT, and decreased GST-M1, what would you need to induce or repress? So for instance, in this individual, they have increased activity of CYP1B1. So what that means is there is rapid conversion of estrogen into a cancerous 4-OH1 form. And that's why what you would want to do is to have a dietary intervention that suppresses the activity of CYP1B1. Similarly, for the same individual, because they have decreased activity of GST-M1, the toxic estrogen metabolites, they are not rapidly converted to non-toxic forms. But essentially, you want to expedite that pathway to make sure these toxic metabolites are eliminated from their body. And that's why you would want to induce the activity of GST-M1 to improve the health of the individual and to reduce their risk of developing breast and ovarian cancer. Now, for the last part of today's lecture, I'm going to be focusing on chronic diseases such as diabetes. So when it comes to diabetes, based on the latest statistics, it turns out that close to 40 to 45% of North American population is likely pre-diabetic. What it means is they are on the verge of becoming diabetic, and of course at that stage they're not going to be going to a doctor because they're not showing the symptoms, and it gets to a point where they're just waiting to become diabetic before they go and see a doctor. And that's why diabetes actually carries a huge burden when it comes to the healthcare costs all over the world. But when it comes to diabetes, it's not a monogenic disease. In other words, just because you're carrying a mutation in one gene does not mean that you go on to develop diabetes. It actually involves almost a dozen genes that interact with each other, and that's how you can truly understand if someone is at risk of diabetes or not. So this is again a very simplistic explanation of how genetics plays a role in diabetes. So let's say there is an individual and they get two different sets of genes. There is a set of genes which makes them hungry all the time. And at the same time, so let's say because they're hungry all the time, they're going and eating a lot of hamburgers, and next thing you know, they're overweight. But at the same time, they have another set of genes which promotes insulin resistance. So because of their because of the first set of genes that they have, which makes them hungry all the time, so they are eating a lot, they're becoming overweight, and because of the second set of mutations which promotes insulin resistance, the overall result is that the individual has a very high chance of developing diabetes unless they change something about their lifestyle. Now, let's say the individual also has a third set of mutation which impacts the activity of the beta islet cells to produce insulin. Then next thing you know, with a lifestyle whereby they're getting hungry all the time, they're eating a lot, they go on to develop type 2 diabetes. But the only point I want to illustrate here is that it's not just a single gene which is playing a role. It's a combination of their lifestyle, the three different set of mutations, which is making them go on to develop type 2 diabetes. So this, this graph here, or this assessment, just this genetic assessment here is taken from one of our reports. And the only point I want to make is that it's over 13 genes that play a role in modulating risk of type 2 diabetes. I mean, of course, we have developed some underlying AI algorithms which look at the interaction of these genes. And if we say someone is at a high risk of developing type 2 diabetes, what it means is that they will have over 85 to 90% chance of developing type 2 diabetes. So what happens if someone is at an increased risk? Well, you got to have an approach that involves diet, nutrition, fitness, detox, and weight loss planning based on their DNA to almost prevent them from developing type 2 diabetes. First, you look at their DNA, you come up with a diet plan, a nutritional plan, a fitness plan, and more importantly, you also come up with a weight loss plan. Similarly, you look at their underlying genetic information to see how it impacts their detoxification pathways, what are the underlying health issues associated with it, and what can you induce or repress to better improve their health. At the same time, once somebody understands that they are at 80 or 90% chance of developing type 2 diabetes, they're very, very likely to comply with the health and wellness plan because they know that if they don't do something about it and they have such a high risk, they will actually go on to develop type 2 diabetes. So final thoughts, if we prescribe non-DNA based health plans, there are three different outcomes. They either don't work, perhaps they work a little in some individuals, and in some individuals you do see a potent response. But if you can cater a DNA based personalized health plan, the only outcome that you're going to have is potent efficacy. And with that, I would just provide a brief introduction to Anam Floyd. So we started around 4 years ago, and our premise was to bring about low-cost, affordable, next-generation sequencing-based predictive genetic test of clinical utility to the masses, and all of our testing is provided through healthcare practitioners. And that's one of the reasons why we have partnered with IHN, because the IHN alumni do understand quite well the role of diet and nutrition, and our testing is based on how do you bring in the genetic information of the individual, and then how do you use diet, nutrition, fitness, supplements to improve their health and also to prevent them from developing a lot of chronic diseases. How does it all work? It's all saliva-based testing. We extract DNA from cells, we sequence the DNA, and then we compare the DNA sequence to our reference databases, and of course, we use our proprietary AI algorithms to do risk stratification analysis. What makes us unique is we have the most comprehensive and robust arrays of genetic tests in North America. Our tests are designed for multiple disorders. We are using the next generation sequencing platform, and what that means is we are sequencing every single strand of DNA over 50 times to make sure we have greater than 99.5% accuracy. And of course, as I said, it's a clinical grade genetic testing, so consumers cannot order this genetic test by themselves, because at the end of the day, it's not just the genetic test. It's about what do you do with that information. And that's why we have partnered with IHN again to offer this course when it comes to understanding genetic tests, so they can incorporate the DNA-based information about the individual and really offer personalized healthcare. So in terms of what we offer, we have a test to look at the risk of breast and ovarian cancer. We have a test to look at the risk of prostate cancer, colorectal cancer, brain disorders like Alzheimer's, Parkinson's, and also autoimmunity. But perhaps for the audience today, the most relevant ones are really the health and wellness genetic tests, because this is more of a high-level overview when it comes to nutrition, diet, detoxification, and fitness. At the same time, we have also developed and launched the world's first genetic test to identify how someone responds to cannabis. Because as I said earlier, even if we were to look at cannabis as a medication, it will only work on a certain subset of population, and a good chunk is likely to see an adverse reaction. And using the genetic test, we can identify who that subset is. Along with that, we have also developed a genetic test for neurological disorders, or what we call the neurogenetics test. And it's looking at the risk of autism, for instance, the pathways that are playing a role in it and how do you modulate it. It's looking at the risk of addiction, schizophrenia, OCD, and a lot of psychiatric disorders as well. And of course, we also have a pharmacogenetic test, which is looking at an individual's response to over 150 commonly used medications. Perhaps, I mean, as I said, the most relevant one of all is the complete health and wellness genetic assessment, because this is really the first step to offering personalized healthcare. Because in terms of what it's looking at the complete detoxification genetic profile. It's looking at the complete estrogen metabolism profile, and for men, it's looking at the risk of developing testosterone deficiency. It's looking at vitamin deficiency, mineral deficiency, a risk of developing type 2 diabetes, high cholesterol, triglycerides, lactose, gluten intolerance, exercise physiology, weight loss planning, and alcohol dependence. And of course, I'm looking forward to getting the course going from March 25th onwards. It's Understanding Genetics for Improving Health Outcomes. And the main purpose of this course is not to make sure that all of you go on to memorize what DNA is. It's about making sure that once you take this course, and if you were to look at any genetic report, you can do a complete interpretation and you can come up with a DNA-based plan for the individual. So once you take the course, you'll gain all the skill sets that you need to offer personalized healthcare. And when it comes to the course, we have 2 assessments. There's a midterm and there's a final. These are take-home assignments, and they're essentially about doing interpretation of some data, which we have taken from some of our reports.
Okay. Hi.
Hi.
That was amazing. So
thank you.
Thank you so much. Your knowledge seems unbounded. I am sure that we have less than 1% entry into that mind of yours. It's just incredible. It's beautiful. I've even had some Facebook messages from individuals saying they couldn't believe some of the things you were saying, even in this 45-minute presentation. So thank you so much. Very honored to have you part of our faculty.
Thank you. It's a pleasure being here. I mean, it's something I look forward to every time.
So if there are no questions, we can say good night for the evening. If anybody wants to ask something in the Q&A that we want to answer, maybe I could send it to you. But
sure, that's not a problem.
I think we're good.
Well, thank you, Julia, and thank you everyone for joining in.
Yeah, very good.
And I mean, as I said, anytime there's a question, just feel free to reach out. I mean, even if you are not taking the course or you cannot take the course for some reason, I think you can get my contact information off the website, or you can just find our email address on Anam Floyd's website and just shoot your questions. I mean, as I say, I'm always based out of Toronto, so you'll find me.
You are very kind, always very kind. Thank you so much. And we'll be seeing you very soon on campus.
Absolutely. Thank you.
Okay. Good night.
Good night.